So, hello everyone, and welcome to our departmental seminar. I am absolutely delighted today. I can introduce a good colleague of mine, Professor Tiina Roose. She is Professor of Biological and Environmental Modelling at the University of Southampton, and she's going to be talking about image-based modelling in biology. I'll give you just a few words of background. She got her first degree in Estonia and then her MSc and DPhil in applied mathematics at Oxford University. She's been a postdoc at the Harvard Medical School, which is where she developed her interests in applying mathematical methods to biological systems. She was also a university research fellow, initially at Oxford University and then at the University of Southampton. And she's been a professor at Southampton since 2013. She works on roots, blood and lymph vessels and lungs, how they develop and function. And as I said, she is principally a mathematical modeler and applied mathematician, but she also designs and conducts experiments to allow her to understand the processes and to inform her modeling. So I'm looking forward to the talk, and I now hand over to you, Tiina. But just one thing, obviously, when we're doing things in remote mode, spontaneous questions are quite difficult to do. But you will notice that there is a question icon. If you click on that at any time during the talk, type in a question. So then we can be a little bit more interactive at the end. Okay, over to you, Tiina.
Excellent. Thank you for having me to give this seminar. I've really been looking forward to it. And thank you, Martin, for kind introduction. As Martin said, I work on plant roots, lung, lymphatics, and blood vessels, and modeling how they function. Now, one could ask the question that what is the joining theme in this, that they're quite different problems. The reality is that all these systems are developed in nature to deliver or remove substances from their surrounding. So plant roots grow in soil and provide plant with water and 13 mineral nutrients. Lung delivers oxygen to the human body and removes carbon dioxide. Blood vessels deliver nutrients to the tissues, and it's the lymph vessels that are very close to, in their terms of their function, to the plant roots, in much that lymph vessels remove the excess fluids and toxins from the human body. So there is a link in terms of functionality. And that link, of course, translates then into the modeling techniques being very similar. But today I will give you a talk about the plant nutrient uptake, because this is the area that has been, by research councils in particular, neglected for a while, but it's becoming now really prominent in the world politics and in particularly when the climate is changing. The motivations for why I work in this area for me have been is that the rich countries use traditionally lots of too much fertilizer, which leads to pollution of rivers and water bodies. But on the other hand, the poor countries cannot afford the fertilizers. In the main nutrient we've been working on is the phosphate, which is there's a lot of it, especially in the UK soils, but it's not available to plants. In addition, it's been predicted to run out in the world, rock mineral phosphate fertilizer resources, in 50 to 100 years. Until then, the most of the mineral resources are in Morocco and China, with only sort of single-digit resources in countries like Syria. So we as a UK cannot rely on these imports of mineral rock mineral phosphate from abroad. We need to have sustainable ways of growing our crops. Alternatives do exist, where you mine the phosphorus from human digestites in the water treatment plants, but these fertilizer products are much more expensive. So no matter which way you look at it, one needs to optimize the plant traits and soil management practices to deal with these increased costs of soil management to be able to grow the crops for human consumption. So our approach in Southampton has been to develop mathematical models to be able to find these strategies in silico in the computer, because you can appreciate the field trials using crops are very expensive, very time consuming, and having fully scientifically based models available to guide the field trial design is really makes a step change in their efficacy. But what is the challenge in modeling anything to do with soil? Well, soil is multi-scale material. It's one of the most complex composite materials in the world, if not the most complex. We have on a pore scale, we have lots of physical and biological phenomena happening, in as much that the soil between soil particles we have air and water, and then we have mycorrhizal fungi and microbes in the pore space. And here you see our imaging using synchrotron X-ray CT, the mycorrhizal fungi in a pore space. The next scale up is a single root scale. So when we grow the plant in the soil, we can now appreciate the complexity of how root is actually living in that soil. This is a wheat root in soil, and you can see the air gaps, which are darker gray areas around the root, which needs to be bridged to the water phase by root hairs and liquid bridges, so that the root is able to actually access the nutrients from the soil pore network. The next phase up is a single root scale, where there are different level of phenomena with the extra microbes that particularly like living near the root. So this is one of our first live imaging deals with synchrotron to elementally map what happens around a single root in soil. And that is sort of live in a sense that it's as close to death as it possibly can. We used X-ray fluorescence elemental mapping to map where phosphorus, silica, iron, and sulfur are. And the sulfur, the green bits, are thought to be a surrogate marker for the microbes in the soil. But ultimately, we want our models to be valid at the crop scale, field scale, because this is where the management decisions are made. So our approach is, I'm a modeler, so we've been developing the first 10 years of my career, we were developing these multi-scale modeling techniques to move from soil particle scale to column scale, plant scale, branching root structural scale to crop scale. And these models are all fairly sophisticated now, but the big problem with them is that they were never calibrated at each scale, and the model is only as good as it is as the data. So we started roughly 10 years ago now calibrating the mathematical models on each of these different scales. And we use the combination of X-ray CT, NMR, XRF, SEM, EDX, and XANES on a pore scale. And by necessity of the scales changing, these techniques against which we calibrate our mathematical models have to change. So when we come to the root branching structure scale, in addition to doing the imaging techniques on a coarser scale, we also use lots of soil sensors to monitor in time-intensive manner the soil chemistry. So whilst X-ray CT and neutron CT and XRF gives us very high resolution spatially on a pore scale, on a big soil sample scale, we go less spatial resolution and more time resolution, because with sensors we can, with microdialysis sensors, we can sample the soil solution at a given location very time intensively, and that again constrains our models very well. So just a little bit of an introduction to image-based modeling here. So X-ray CT works by taking X-ray pictures at many different angles. So if you broke your arm, you go to the doctors, they take X-ray from maybe 2 directions to determine that your bone is broken or fractured. In the high resolution X-ray CT, you rotate it through the X-ray beam over thousands of very little angles, and then you reconstruct the image to see the 3D image of what you've just imaged. The following acquisition of the image, we then segment it out. So we effectively get the structural information from these images about the root morphology and the soil particle and air morphology. Now that would just stay as a pretty picture. It doesn't necessarily say very much about functionality of the sample you've imaged. We add the functionality by doing the image-based modeling on those scans, and then guide it further for the next scans that are necessary to be done by using modeling to make the models better. So our classic pipeline that we use is that we image, we then segment, and then we go 2 ways in terms of morphology modeling. One is we do direct simulations on the images we require. So we generate 3-dimensional mesh. We use our supercomputer and do the modeling. Now that's the time-intensive bit in the stream, but in parallel, we also extract out of the images 3D morphology and 3D morphometry, which is faster, but bringing the 2 together in the end, in terms of 3D modeling and fast morphometry, we can assign structure function associations to our images that we acquire. So I will give, after this brief introduction, now a little overview of various different projects we've done. And the first one I start is the project that was funded by BBSRC, where we looked at how barley roots, when they hairy or hairless, grow in soil. So we did pore scale imaging with synchrotron and modeling of barley roots in soil, and then we fed within that same project this same information into the field trials that were done at James Hutton Institute in Dundee. So the root hairs are these single cell extensions from all root surfaces. Most roots are thought to be hairy. They are thought to be important for strongly bound nutrients like phosphorus, so that because they penetrate these micropores, so they can access more phosphorus than bold roots. One can breed hair roots with current breeding programs to be more hairy or bold, you can make them longer, shorter, branched. So the question is, what should we do if the climate is changing to be able to have less fertilization, but have the barley still grown and not lose their yields? So the first thing we did with these hairy root problems is we took the idealized geometry. So we assume that the hairs the roots are completely hairy, they nicely regularly hairy. And then we used multi-scale homogenization techniques, which basically is a zoom up technique from going from small scale modeling of a single hair to a collection of hair. And in that process, we determine which phenomena are important on a root scale. To recap how that roughly works is that we separate the scales. So we have macro scale, which is then the soil scale, length scale, and then we have micro scale, which in the case of root hair is the size of the root hair. We write down the model on all scales, but obviously when asking questions on a field scale, there's no point in modeling every single individual hair. So we then take this model on all that is valid on all scales, and we use asymptotic approximation to find the solution, assuming that these two macro scale and micro scale are separable quantities. We collective successive powers of our perturbation expansion epsilon here is now the ratio of this micro scale to macro scale. It's a small quantity. Collecting the different powers of our perturbation expansion, we arrive at the macro scale equations, and I've obviously brushed a lot of mathematics under the carpet here. But the long story short is we find there are three different models that could explain how root hairs function for phosphorus uptake. And one is negligible root hairs. That means the root hairs sit there like little sponges in a soil pore space, but they don't generate the diffusion gradient because diffusion is faster than the rate at which they are absorbing anything. The second one is dynamic hairs. So that means that the root hairs are taking up phosphorus at roughly the same rate as the diffusion is resupplying it. And the third one is that they are like little hoovers in a pore scale. So they as soon as the hairs go into the pore space, they hoover up everything, leave nothing behind. So when we went and looked at the literature, we found that actually most of the literature suggested that it's these little hoovers. The root hairs are little hoovers, so they take up all the nutrient as soon as they see it. But we also noticed that the root hair morphology was always measured in a petri dish, so there was no soil around it. And now you can imagine very easily as an engineer that actually root hair growing effectively in the water would develop a different morphology from the root hair that grows in a soil pore space where you have soil mineral particles on its way and it has to sort of meander past it. So we really needed to look into imaging and finding out what are the root hairs doing in the real soil. Now the challenge for imaging and seeing into soil is that the root hairs are roughly 10 microns in diameters and they 100 to 1,000 microns long, so they long thin structures. And to be able to detect them, we need at least 1 micron resolution. Now, tabletop X-ray CTs can do this resolution, but that comes at the expense of scan time, because the scans can take 20 to up to 20 hours to do at that resolution. In that time, plant roots grow a lot because plant roots actually grow 2 centimeters per day. So you can see all these shadowing effects. So we went and saw, did this imaging in synchrotron beamlines, and you saw the movie on the right here of the assay that we used to be developed to be able to do this imaging at the synchrotron beamlines. So this resulted in rather pretty pictures, but as I said, pretty pictures stay as pretty pictures until you assign some function to it. And we did this assigning of function to the morphology by running them simulations directly on these images. So we run the diffusion equation on the pore space with assuming that the uptake of nutrients on the root and root hair surfaces is given by Michaelis-Menten uptake, and then assigning the first order kinetic reactions, binding reactions to the soil particle surfaces. So this enabled us to then predict how much phosphorus this root and root hairs are taking up, directly relevant for these images. And what we found is that the root hairs actually take up, the effect is to double the uptake of nutrients by the root, rather than be they not these little hoovers that go into pore space and dominate everything that was previously thought. Here's on the left the simulation that shows this prediction on 3 different agronomic phosphorus levels. So phosphorus level 1, 2, and 3. 3 is where UK government suggests not to fertilize that soil anymore. In all cases, it was the balanced pathway needed. That it's the model 2. It isn't the little Hoover model. It's the diffusion is roughly the same as hair uptake model that we need to translate to the root scale, to the field scale. The other bit that obviously the image-based modeling allows is that it allows us to quantify now this local effect for phosphorus uptake. It's intuitively clear to anybody that if the little fine fibers are growing into the soil, they would take up phosphorus more locally from the very localized pore spaces that they inhabit. But with models and imaging, we can now say how local that is. And again, as we see that the root itself is scavenging phosphorus from roughly double the area of soil particle surfaces compared to the actual hairs, which take up half of, only access half of this area of soil particle surfaces. So moving on from there, we also knew that the root hairs are known to exude surfactants effectively and chemicals to the soil that changes soil water behavior in the region of soil that roots that is directly surrounding the soil. So we wanted to know what that effect is. When we did the mechanical tracing of soil particles, and you see here on the right, we found that around the root we have an increased porosity when we have the hairy roots, and then there is a dip of porosity further away, and then it recovers to the as a distance. It recovers far away from the root, back to the bulk porosity. So we wanted to know what the effect on water movement from this soil mechanical particle movement due to root having grown into soil is. Because that determines what that pore structure change means for plant water uptake. Now, soil models classically for soil water uptake models is called Darcy-Richards equation, because it combines the Darcy flow for water with two experimental equilibrium measurements, and that is, one is soil water release curve that relates the soil pore water pressure to the relative saturation in soil, and experimentally measured hydraulic conductivity, again, which needs to be measured for each relative saturation level separately. Now, these experiments, to be done accurately, are very long experiments. In addition to the hydraulic conductivity and soil water release curve, exhibit hysteresis. So that means that the wetting and drying paths are not the same, and these need to be measured explicitly for each microstructure. Now, this is clearly quite a big undertaking for conventional soil measurements, but more importantly for roots. We can't actually extract the soil from around a single root and do these experiments because of these microbial transformations in the soil. The sample in the beginning of experiment would not be the same as it is in the end of the experiment when you're doing wetting and drying, because the microbes will be born and dying around it. So we needed to find out the way from estimating the soil water release curve and hydraulic conductivity directly from images. And we did this by developing a workflow pipeline for modeling Richards equations based on images. We used what's called phase field modeling of the soil of the air-water mixture, and then applied these two-scale homogenization methodologies to this. And what you can see here now on the right, we can predict these air-water, the soil water release curves based on images, and more important, we can manipulate the important parameters in it that might induce the changes to the water behavior in soil. So normally when we assume now that the bulk soil has contact angle 0, so all the soil particles will always remain wetted with water as you wet and dry, there's very small hysteresis loops as a result of this in the soil, and that's been compared to experiments and fairly well known. But when plants exude organic substances to the soil that change this contact angle by 20%, we see that the hysteresis loop in that rhizosphere changes dramatically. So it becomes much more chaotic, and the wetting drying behavior that has major implications on plant water uptake. So having done these images, we now brought together these mechanical and water modeling and started looking at what that has what impact this has on the chemistry of soil. And remember, we had around the root the increased porosity zone, and then compression zone and bulk soil zone. And what we found, to cut again the long chemical X-ray mapping story short, is that actually the root having grown into the soil and creating this increased porosity zone and then compression zone further away, that compression zone actually physically seals off the microbial active zone around the root, so that all the microbial rhizodeposits and nutrient uptake and microbial transformation remain physically constrained into the immediate surrounding of the root in the increased porosity zone, and they can't escape very easily through that compression zone into the bulk soil. So we call it now a root microbial incubator zone around the roots. Having studied now in great detail what happens to these root hairs around the root in detail on a very small scale, we then translated these to the field scale situation and compared it hairy versus hairless with different irrigation scenarios, with different phosphorus profiles in the soil to determine what the field scale impact of hairs is. But I want to now move on from this field trial results, which are still yet to come in, to explain also how we're using the same approach in modeling, for example, slope stability. So in England, we had an issue roughly 20 years ago that the trains were delayed because of leaves on the line. That problem was solved by chopping down older trees off the embankments, which resulted in a different problem, that the railway slopes became unstable. We have now slips on railway embankments. So we started looking at how the plant roots influence the stability of slopes, and to model this. Now, the plants stabilized soils in two ways. The most intuitive way is to think that they are mechanically in the soil as a fibers, and they make it, hence make the soil stronger. But the roots, even if the roots are very fragile themselves, they actually strengthen the soil by being in the soil and sucking out water. So they maintain the pressure inside the root that is sucking the particles together, and it's the combination of the two that makes the stabilizes the slopes. So we've developed the mechanical hydraulic models for dealing with the soil stability problems. It effectively is the dominant mechanism we are finding at our preliminary studies at the moment here is that it is actually that sucking of water, maintaining water tension, that is the dominant mechanism for slope stability, rather than necessarily the actual fiber reinforcement. But the jury is out on this one yet. The next bit I want to talk on a sort of slope or field scale is use of microdialysis probes in experiments. So microdialysis probes are these little sampling technique for determining what's happening in a soil poor water. We send in dialysate and then perfusate and then measure what's coming out of it. And in the membrane section there, we can also make those probes as a little mini artificial roots. So we can deliver chemical compounds to the soil and see how soil responds in a much more controlled manner than just using a sort of root as it is. And so this is what we did with trying to understand how roots exuding citrate influence the phosphorus uptake. So we delivered the citrate solution via the microdialysis probe and measured how much phosphorus came out. This is what roughly plant does, but plant exudes, in addition to citrate, all sorts of other compounds into it as well. So with this careful technique of microdialysis probes, we were able to characterize for the first time a very complex model for soil phosphorus and citrate interaction in a soil pore space. I'm not going to go and describe you all the different terms here, but we took into account the microbial turnover of organic compounds delivered by microdialysis probes. That, to cut the long story short, we were able to carefully, all these uncertain parameters in the models, which are shown here in red, we were able to independently calibrate them by using these microdialysis probes, by doing the set of 5 different experiments to extract all the different parameters in a controlled manner. I think it sounds very simple, but for the plant-soil interaction area modeling, it is really important to start looking and constraining models very carefully against the data, rather than building ginormous models which have lots of different parameters and that are calibrated against 3 data points. There's too much freedom, so our task here was to calibrate the model against the data which was constrained and well defined. And the microdialysis probes allowed for it, so these are the results for fitting. And the conclusions from this work were that the citrate enhanced a lot the phosphorus uptake and solubilization in the soil. We got up to 10% gain of phosphorus extraction in a very low phosphorus soils, and the root system architecture is very important to achieve these gains. Because this root architecture is important, we're now working on determining what particular traits one would need to breed into the plant root architecture in terms of not only architecture, but also citrate in exudation to be able to grow crops in a low phosphorus conditions. I want to finish this talk by just showing you a very recent work we did, which is to do with actual looking at the fertilizer granules and their behavior in soil. It's been anecdotally from farmers we've heard is that when you fertilize the soil, the microbes in the soil respond, and some of them think that they respond by creating these microbial blooms, and the other ones think that actually they can die, that your microbial diversity in the soil reduces the more you fertilize, especially when you're doing nitrogen fertilization. So we took a fairly standard, agreed, well-parameterized nitrogen model, and then put it on to the image-based modeling framework, where we have on the left bottom corner here fertilizer granule that is releasing nitrates into the soil, and then we see how it moves in that pore space and transforms into ammonia and organic compounds, nitrogen compounds. We then did experiments to see how microbes would respond to this. And what we found was that actually, as the fertilizer is releasing these nitrogens, the salts into the soil pore space, what we see is a death zone. So on the right, you see the red is the death, or when we death zone of microbes, which we define as a 10% of microbial activity has dropped from bulk soils 100% to a 10%. And that size of the death zone is heavily dependent on soil water status. So in the high levels of water saturation in the soil, the death zone doesn't really occur, although you get a reduction in microbial activity. Because of this, salts are not accumulating in a soil pore space, they get diluted. Whereas when it comes to lower saturations, the dilution effect is dramatically reduced at lower saturations, and you can get these microbial major death zones around the fertilizer. So by fertilizing soils, we can disturb quite significantly the soil microbiome and kill off major bacteria in it. We've also looked at the models on a field scale now, going on from where we look at the ridge and furrow fertilization of things. So potatoes are done in a ridge and furrow system. The current safety standards say that the ridge cultivation is much more environmentally unfriendly than the flat field combination, because they say that the runoff from ridge and furrow can be bigger. So we developed the models for this, and what we found that actually that depends on the rainfall pattern, how much solutes get leached from ridge and furrow compared to flat soil. So if the rate of rainfall goes higher or is closer to the ridge and furrow, to the fertilization event, the runoff is much bigger in a flat soil, in a ridge and furrow, than in a flat soil. But when you have high rainfall events, the flat soil is actually much more prone to leaching than the ridge and furrow soils. I'm conscious of time here, slightly. We've also done so in the root area. It's really trendy to use X-ray CT scanning and build these water uptake models based on imaging. Now, this is actually our example of where we did a negative control. In a sense, we compared our multi-scale image-based model that is one-dimensional, what is running in the middle domain here, with scanned image-based all singing, all dancing model of water uptake. And what you see here is that our multi-scale methodology actually agrees really well with image-based modeling result. Now, the caveat is that our multi-scale model that is one-dimensional is very fast to run, so we can run lots of different scenarios, whereas image-based model is very labor-intensive and takes lots of computation time. For water uptake, our conclusion actually is we don't need to do imaging at all if we want to do plant water uptake only, because the difference in the two models is sub 2%, so it's well below any experimental error. And this is a clear illustration is that if you're interested in plant water uptake, in my opinion, you're wasting your money for scanning. You need to have some other reason for doing expensive X-ray CT scanning than just predicting how much this plant takes up water. Fieldscale, we've looked at different architectural scales here, where we look at should we breed the plants that have more branches up, higher up, so exponential branching or linear branching. It's undoubtedly clear now that this just breeding a plant with better root architecture can recover the same amount of phosphorus from P1 soil as the bottle brush plant root system is extracting from P3 soil. So really we giving the scenarios for plant breeders now to help us with reducing the need for heavy fertilization that's been going on in all 20th century. We'll go for, so conclusions I want to move on to. Why should we be doing image-based models? They are really quite labor-intensive models to do. In my mind, these are the alpha models. They enable the testing of the most detailed science to be done, but they also allow us to validate the faster upscaled models and thereby build confidence in model building exercises with the domain experts, because domain experts often see the details being important with doing both the model, which includes the details, and then does the multi-scale faster model. That is excellent tool for showing building confidence in overall modeling processes, and I think that is important for take up of these models by the plant and soil and environmental community. So I want to finish here. I have a couple of more slides that I can put on later, but I obviously don't do this work on my own. I have an excellent team of postdocs available and a number of very close and dear collaborators who from soil science who keep me on straight and narrow that I do really good work. And these are David Jones from Bangor, Guy Kirk from Cranfield, Philip Hinzinger from Montpellier, and Doris Wetterlein from Leipzig Helmholtz Center. So I want to end here and take questions.
Okay, well, thank you very much, Tiina, for an absolutely excellent talk. I'll give a little round of applause there. So now we have just under 10 minutes for questions. I'm going to take sort of chairman's privilege by asking the first question. You obviously the title and everything is about image-based modeling. So you have an image.
Yeah.
What do you then do? Do you segment the image into, say, pore space and solve equations in the pore space? But then how do you do it when you have the roots and the small roots going into the soil? I wonder, can you give us a little bit more detail in exactly how you do the image-based modeling?
So that depends on really the scale of a problem that we're asking. So when we do the root single root scale root hair problem, for example, we do segment out hairs separately from root surface, from the soil particles and air-water interfaces and the pore space. We then, from segmentation, we used softwares to generate the meshes for computation. We use ScanIP for this mesh generation because we find that's really useful. And then we run on those meshes the numerical simulations of the models directly by using either COMSOL multiphysics or not Interpore. What's that package that is out of Imperial developed?
OpenFOAM?
OpenFOAM, yes.
Yeah.
To run the simulations. When we're doing so that basically locks down us the microscale, pore scale models for us. And we then, when we're doing a root architecture scale models, we only segment out root architecture, and then we apply those pore scale models that have come from pore scale onto the root architecture. So in root architecture is the only thing segmented out in that case where it appears as a boundary condition. So one thing we have avoided doing a lot is averaging the root density. We want to really know what the root architectural traits are. So we segment out the root architecture, validate the models, apply the pore scale models that have come from that other scale of image-based modeling to those root architecture models, and run the simulations. Again, ScanIP, COMSOL, that tends to be only COMSOL. There is another layer on top of this where we also then use the averaging out the root system. So that isn't image-based as such anymore. Then we take the architecture that we've scanned or imaged, and in that case we can use X-ray CT images, but we can also use, there's lots of imaging data coming from shovalomics experiments, what's it called, where people dig out those root architectures and then optically image them on the field. That gives us the root density, and then we take the image-based models, average them, and then run the distribution-based models effectively.
Okay, thanks, Tiina. That gives me a lot of information. Interesting about the meshing. You have another question here, actually, which is moving now on to the average models. So this is from JP Latham. You mentioned for the water uptake model that you felt the image-based model was not necessary. Yeah. Okay, because you had this average model that worked. Okay, but what input parameters do you need in the average model, right? And how do you obtain them?
So for image, so if you want to do a water uptake model that is image based, you need to image it and then run it directly. If you want to run the faster model that I showed equally accurate, you need to know the root length density distribution, and there are faster and cheaper methods than X-ray CT in that case to determine that length density, like shovelomics techniques and wind riser techniques, which are quicker and give you the necessary information, and they're fairly easy to implement. You know, CT machine, you can't take it to the field, but the shovelomics techniques and the optical techniques that they do when they pull out those plants, they can be done on field trials. In US, they get done a lot. So you still do need root architectural information, but you can get it much faster and cheaper with other techniques to validate these plant water uptake models.
Okay, yeah, I can see some other questions. I'm just wrestling with this. So one comment we have, and I understand this because we're looking at rocks, right? Mm-hmm. And there's a big contrast between, say, rock and the fluids within the rock. But even then, with the fluids, we have to dope the fluids in order to get ideal contrast. So the question here is, how do you enhance the contrast between the different objects in the images, say between hairs and roots, or even between that and the water? I mean, because fundamentally there seems to be a lot less image contrast than we see in our applications.
So in hairs, we didn't have to do any contrast enhancement. It was fairly with synchrotron. Obviously, you get very high resolution images and good quality images. So we didn't do any contrast enhancement there. We have looked at the contrast enhancement in detail, where there are basically, we've used medical contrast agents. We've done a whole scan. Some of the medical iodine-based contrast, X-ray contrast agents for root imaging have performed as excellent herbicides.
Yes. That's the problem, is they don't mess up rocks, but they'll mess up plants, right?
Yes, they mess up plants, but some of them actually do work quite well. The way we deliver them to the plant is there's we cut the stem on top of the soil column and dip it into the contrast agent, and then by phloem sort of plant circulation. Plant has a circulatory system like humans have. It's slightly different because it doesn't have a pump, but plant circulation will transport the contrast agent down into the roots. So we've looked at it mainly from a point of view of accelerating this segmentation process, because we can usually see the roots. It's just by hand segmentation is really quite tedious and labor intensive. Whereas if we deliver the contrast agent via cut leaf from the top, it makes the segmentation sort of semi-automatic, fairly automatic. So that's one purpose for us. We're also looking at, as you know, we have a grant pending with you actually on this, using gold nanoparticles to do the pulse trace of water movement in the soil from the soil side. The problem with iodine-based contrast agents in general is that you don't want to apply them to the soil side either, because they kill the soil microbiome. So gold nanoparticle-based contrast agents are really good at using on the soil side, so that you could ideally do the pulse trace experiment in soil to see where the water is flowing to the root. So there are different techniques one can look at when doing the contrast enhancement. This is an open field. There's lots more people needed for this to make progress. Because I also think, in addition to these nanoparticles, the existing medical contrast agents, they've obviously been optimized for human use. But I think there's some work to be done to optimize it for plant use, the formulations, to enable further work in this area.
Okay, thank you. I think unfortunately we've run out of time now. We do try and keep the seminars to the hour because lots of the people listening have other commitments. But thank you very much, Tiina. Really interesting talk, and it opens up a whole range of applications for imaging and modelling that we do in this department, but to other very important and fascinating problems. So really thank you very much once again. We'll just end quickly with an advertisement for next week's seminars. Next week's seminar will be Professor Inge Behrer from the University of Bergen, which will be talking on modelling injection-induced coupled dynamics in faulted formations. So I think we'll all look forward to seeing everyone again this time next week. So goodbye.
Goodbye.